I'm going to introduce now Yana, who will introduce the next speaker, which is Betul Kaçar, if I can pronounce that correctly. Um, so Yana got her undergraduate degrees in biological engineering, computer science, basically, and biology at Stony Brook University. And I'm not sure, Yana, did you have Masiori as a professor?
I did not. Now I feel bad about it.
Okay, so Masiori, I think, was a professor and probably chair of the department when you were a student at Stony Brook in biochemistry.
He left in 1984.
Oh, so way before you were born. I'm sorry, Yana.
All right. Well, I don't feel as bad anymore.
Okay. Anyway, Yana then went on to do her PhD at Columbia University, and she did a postdoc at Columbia. And I met her on a train on her way down to an NIH conference in Bethesda. And the next thing you knew, she is hired at Rutgers University. And she is now, I think, a full professor.
I am. Yes. Thank you.
Mazel tov. Okay,
Thanks.
so Yana, it's my pleasure now for you to introduce Betul.
Thank you, Paul, and thank you for bringing me to Rutgers too. It's my amazing pleasure to introduce Betul. Betul Kaçar is an assistant professor of astrobiology at the University of Arizona with appointments in now 3, as far as I understand the departments of astronomy, planetary science, and molecular and cell biology. So this is really great for someone who has been associated with 2 departments with people going, oh, that's crazy. Now people must be really impressed by what you're doing as well. I know I am. She's also an associate professor at the Earth Life Science Institute at the Tokyo Institute of Technology. Betul received her PhD from Emory in 2010, working jointly in the Department of Chemistry at the Emory School of Medicine. Since then, she was appointed as a postdoc at Georgia Tech and later awarded a NASA Astrobiology postdoc fellowship in 2012. Between 2014 and 2017, she led an independent research project at Harvard University's Organismic and Evolutionary Biology Department. In 2017, she joined University of Arizona, where she's now focusing on reconstructing key enzymatic intermediates between biological activity and global geochemical reservoirs throughout Earth's deep history. She directs a NASA Astrobiology Research Center exploring the essential attributes of life and its origins. And she's been recognized by quite a few awards, but one I really want to bring up. In 2020, she partnered with the 2020 UN Women Generation Equality Campaign to support education of girls and women globally, and delivered a talk during the UN meeting for the Commission on the Status of Women in 2021. And I find that absolutely amazing. So thank you, Betul, for taking that on. And thank you for accepting our invitation to speak.
Thank you so much, Yana, for having me, of course, to Paul for the invitation and for this fantastic organization. So I started my wet lab. I set it up in January 2018. I remember that well. And so we've been since trying to resurrect the sort of ancient proteins experimentally and do a lot of work computationally as well. So there are really nice overlaps between what we do and the ENIGMA work. And so this was, I think, sort of meant to be. So finally, we got to meet, even though it is through computers. Before we get started, I also want to give a quick shout out to Don Tevfik and remember his memory. So I've never actually got to meet Don in person. I was looking forward to this meeting. In fact, when I received the invitation, I thought, oh, finally, at least through Zoom, we will get to meet. So unfortunately, that didn't happen, but I still wanted to just share a very quick sort of memory I have with him, of course, aside from his wonderful papers that definitely shaped the way we think as a group. And we always refer to his papers, and we still do say what Don thinks about this. Let's see whether he wrote something about this. But back in 2017, he attended a talk in Turkey, in the Middle East Technical University, organized an evolution workshop to remember a prominent evolutionary professor, Aykut Kence. So what made this special for me was that it was a year, particularly difficult year following 2016, a lot of explosions in Turkey, a lot of conferences being canceled, and a lot of people canceled their attendance as well. But Don was the only professor, not only didn't cancel his talk, but also gave a keynote and personally came and delivered this talk. And it really meant a lot to me and many of us studying evolution in Turkey. And really, I think delivers the point that what we do beyond our own ideas are, of course, wonderfully competing to get better and better, has broader impact and meaning and what it really means in the global context. And I think Don was an example of that. So I remember him fondly, and I will continue to. The work we do sort of is all about connecting scales, right? So we try to connect molecules, particularly proteins, and their interactions in the subcellular level to what happens at the planetary scale. This was, I think, inevitable that we will at some point come to this point. For decades, we've been in a way reducing our ways of analyzing what's happening in the cell, while also trying to broaden our way of understanding them by connecting them to evolution, which I think is big as it can get, really, when you try to understand life. But astrobiology especially is, I think, pushing biologists to think even broader, that what does the sort of molecular interactions that we're trying to understand mean within the context of planets, and whether we can understand, even let alone quantify, what the molecular interactions mean and what do they do in impacting the global scale observational properties. That's, of course, a very bold goal. NASA recognizes that, and we are all here because of NASA's vision, right? So, and we tend to think about life, but whatever we analyze around us today. So we have this problem in astrobiology that we say, okay, n equals 1 problem, there's only one form of life. So whatever we look for feeds from what we see around us today, that's beautiful, but also restricts our ability because we don't have much diversity. That is not true, of course. Any microbiologist knows this, right? So we know that there's incredible diversity of life that we see today, and life adapts to its environments in the most simplest way. And I'm putting in the most simplest way, of course. So when we think about the past, and when we think about the past of our own planet, and when we realize that the environmental conditions in the past were vastly different than what we have around us today, that inevitably opens us this idea that there's not necessarily one form of life, but there's all these other niches that were present in the past of our planet, and biology and biological diversity that responded to those niches. And we want to understand what happened then. So in a way, this is our own way of sort of trying to understand the past to increase our own sample size to n equals to a little bit more than 1. And I will drill down to what this means throughout this talk, and I will also focus on nitrogenases, which is actually, it was great to hear Douglas Pike right before me. So I'm hoping that there will be a nice organic synthesis, not just between my talk and his, but also several speakers before me, even the ones earlier today. So then the question is, let's deduce it down a little further. We're looking sort of the role of contingency and determinism, but in the sense of understanding mechanisms that impact biology. So the question is, given an alternate planetary environment, would we still find an Earth-like life? And of course, we need to keep our minds broad, that Earth-like isn't necessarily too narrow as we might think or fear. And of course, with other planetary environments likely enable the emergence of the metabolisms and the processes that we see that sustain our Earth biosphere. So this is not an easy problem, right? There's probably no smoking gun that we won't be able to look at a planet and be able to understand whether a metabolism that is familiar to us exists there or not. But I believe in the next decades, maybe century, this is going to be what we will try to do, and I think we will get there. Definitely this is a great time to study these problems. And my lab looks into this. We have 2 main challenges that we're trying to tackle. One is the origin of life. Traditionally, those familiar with the origin of life field may think of this as a bottom-up problem that start from raw chemicals and even subatomic particles in some cases, and try to understand whether they form some meaningful networks, meaningful in the sense that whether they would resemble any metabolism or a network that is useful or familiar to us by our understanding of biology. That's not what I'm going to talk about today. And the other part that we study is the early evolution of life, and in some cases the earliest. What happened once life emerged? That's really when the party starts, right? So we want to know what happened, what the sort of interactions that happened between molecules, what was there? It's a quite remarkable mystery. We use tools that are very familiar to most of you in this group to reconstruct the biology of early Earth. It's a mix of computational studies and lab work. We try to resurrect ancestral biosignatures, and that's what I'm going to talk about a little bit today by discussing the example of nitrogenase. And the overall goal is to, again, see whether the possibilities of life to what we know exist, and also how much can we alter life that we know to in a way that maybe is a little different, even though it's still building on the molecular architecture that is familiar to us. And of course, ultimately assist finding life beyond Earth. So when it comes to biosignatures, or so when we mean by life, of course, now scientists, they had to quantify this in some way, so we reduce it down to biosignatures, and there are different types of biosignatures, and there are a few really nice review papers that came out in 2018 reviewing these biosignatures. If you're interested in them, they were published in the Journal of Astrobiology. But the idea here is that when it comes to, especially if you're interested in experimental work, I think, or the more important biology angle, we want to understand the sort of microbes, and which they are likely the dominators of the past, and of course the current state as well. And here's a nice quote from Tim Lyons and Aubrey Zirkle, that when we ultimately find a biosignature for life beyond our planet, it's most likely going to be microbial. This is a very strong assertion, but there's a reason why we think like this. And again, it comes from our understanding of life on our planet. And the way we study past as a collective, it seems like in this astrobiology group, and those folks who may not necessarily call themselves an astrobiologist, but really exploring these things at the same time, is that our, I think, remarkable work that is done by decades to integrate the molecular record and the geologic record. And as biologists come up with these wonderful tools, computational tools, to figure out how proteins evolved, and yes, maybe they are discovered to study a cancer or different diseases with direct applications to human health. We extract them and we can apply to them the questions that involve deep time evolution as well. And that's what's been happening. So we have the molecular records. It starts by understanding the extent molecular diversity. We use phylogenetics. The field uses molecular clocks and try to constrain the timing of the emergence of essential metabolisms, and whether we can shift our understanding of when, say, oxygen appeared on our planet and whatnot. So that's one record to understand the past. And the other record comes from geology, and we get a brief presentation to this today as well. We have fossils, biomarkers, a lot of tools coming from geochemistry, biogeochemistry, remarkable work in these things called clumped isotopes. Even the name sounds so sophisticated and complicated, but we have all these tools to also understand the geologic record. And we've been really trying to integrate both of them based on the understanding that life is shaped by environment. So let's try to integrate them and understand what's really going on. And at the heart of these two data sets really comes the molecules. Even if you're thinking of microbes, it is at the end of the day, the metabolisms and everything you see are mediated by them. So this is why most of us are passionate to understand the enzymes, because they are the mediators between life and the environment. And this is true for ancient life as well. And this is true for understanding ancient biogeochemistry as well. That is the ultimate integration between biology and the environment. So you're looking at a sort of a rather hypothetical enzyme here with different metabolisms. And on the right, it's a paper that we often visit in the group that really draws on the point of what's going on between the earth mantle and crust, and as well as the atmosphere. So I want to put the enzymes between these two records that come with their own problems as well. So of course the story isn't all very sweet and easy. That we have the molecular records, it's great, but of course most of the things are probably overwritten, right? We know this from geology, that probably what the number is, say, I will be very generous and I'll say 5% but it's much less than that. Or whatever happened on the history of time is wiped off, right? So we don't have this record, and we sort of learn how to live with this. But we acknowledge the problem here, that the diversity, especially the molecular diversity or whatever we grasp is from studying today's biology, is a very limited picture of what actually happened. And that, I think, is also what makes this field very exciting. That you really need to have a little Sherlock Holmes hat when you're trying to explore what may happen in the past. Even though we have the limitation of the molecular record, we also have the limitations that come from geologic record interpretation. That, of course, the process of fossilization is rare. The record gets spottier and spottier as you go back in time, especially if you pass the point of, say, 3 billion years. You have maybe a handful of samples, maybe, and our understanding comes from those. So that's also mind-blowing, inspirational, also terrifying. That whatever we study is also not naturally selected, unlike the molecular record. They have to be found, and they have to be preserved well, and they have not to be degraded. So these are the problems that we accept, and that's also why I think enzymes are neat, in the sense that, you know, we tend to think that whatever exists today are mostly essential, and they're most likely to be ancestral, and whatever is preserved amongst these 3 domains of life likely to have existed in some form in their origin node as well, as maybe you can even trace as back as the last universal common ancestor. And enzymes themselves catalyze these metabolisms whose output can be read in the rock record, right? So say a biosignature that may be found in some fossil remnant is ultimately present because of a metabolism, and that metabolism was catalyzed by some enzyme whose offspring exists today. So understanding today's life and how these enzymes give rise to these distinct biosignatures is important, also to understand the enzymes that may have existed in the past. Yet our connection between these 2 data sets, in my opinion, especially with the integration of enzyme evolution, is quite limited. That the paleobiology isn't quite integrating the molecular scale evolution in interpreting these biosignatures that are found in the rock records. We are not there yet. And nitrogen isotope is one of them. And we've been doing some work with those, but as well as the carbon isotopes, by studying the proteins that give rise to the distinct biosignature record. It could be even as preserved organic matter form or whatnot, and not only nitrogenase, but we also do some work studying the enzymes such as Rubisco. So then we can argue that evolution of enzymes and understanding them thus opens us to understanding the other record of life, and really overlap the earth evolution and enzyme evolution is what our goal is here. That how has the environment driven the evolution of enzymes over Earth's history? And I'm talking billions of years of time. I'm not talking about going to a permafrost and extracting, maybe finding some fossilized ancient microbe and analyzing their protein behavior or DNA behavior. They're still too young for us. We are interested in the deep time. And also vice versa, of course. How did the environment impact the enzyme evolution in return? And I refer to this approach as the molecular paleobiology. It's a pretty integrated tool. I see some of you smiling. Yes, I know you're probably familiar with Kacar. Of course, day-to-day activities are less boring than that. But we are synthesizing a lot of different tools in my group. Everything that we do is shaped by evolution. Everything that we do, always we visit over and over again, how and why, and then try to take a step back and see the broader picture. And I think evolution gives us that. So we do a lot of phylogenetics and experimental evolution of ancient DNA and ancient proteins, more specifically in my group, and try to understand the gene and protein level interactions by engineering these ancestral DNA components inside modern microbes, and then studying the output generated by these microbes in terms of their biogeochemistry, or as simple as their fitness or growth properties, to then connect to them to interpret or revisit the environmental level behavior that is coming to us from the geology. So this is really a synthesis of synthetic biology, evolutionary biology, and geobiology to understand early Earth biology. And we focus on different systems. So you're looking at the timeline, the formation of Earth. We have a rather quick stabilization of the atmosphere, so origin of life somewhere around here. So say depending on the results that you are on board with, but let's be conservative and put it around 3.8, that is the origin of life, so that everyone is happy. And then we have some understanding of what existed in Luca. Of course, this is coming from a lot of work that also was carried out at Georgia Tech, that looking at RNA, looking at ribosome, try to understand the first molecules of life and understand the first machineries of life. So in my lab, we do a lot of work trying to understand the translation machinery, so sort of making it a little bit more complex than ribosome itself, but also integrate the proteins that are functioning, they're assisting the ribosome, and understand how they evolved. And we have a paper coming also to start drilling down in addition to the one we published last year. We also study different metabolisms, not necessarily, we're not necessarily interested in understanding their when did they necessarily originate, so we're not really constraining the time, although sometimes that it's very difficult to step away from that. When the data tells you something, you have to read it. But we try to understand the evolution of nitrogen fixation and carbon fixation by resurrecting ancient Rubisco enzymes and inserting, we engineer them inside modern cyanobacteria. We actually have a preprint that came out last week where we resurrected an ancient Rubisco enzyme inside cyanobacteria and measured the biogeochemistry generated by the cyanobacteria that is imposed by the Rubisco. So we're trying to really create this ancient carbon isotope record using these engineered synthetic systems to see whether, first of all, we will be able to replicate what is thought to be the case in the past, and if we cannot replicate, why? So it's pretty interesting questions here, but we are also interested in looking at the proteins that are involved as photoreceptors and looking at their evolution as well. And all of these things really feed from the sort of the experimental tool set that I presented early on. And we are also doing some experiments. In a way, you may think of it as, if I may say so, it's sort of a Miller-Urey experiment, but in this case, using radiation, and see how we can generate chemistry and complex chemistry using radiation as an environmental source. And most of this work is carried out in Japan. So going back to the sort of our own moderate problems, which is time travel, the challenge is that how do we reconcile? How do we go from ancestral DNA? And of course, I'm going to be telling you about how we go to the ancestral DNA. But how do we then go connect DNA to the enzyme organism ecosystem to planetary scale? So this is a big problem, even though life does this. Right? So, but how do we do this in the lab and create these experimental systems while engineering different scales to reflect the past? And also, how do we interpret and make what we have and make sure that what we see is imposed by the ancient DNA and not something else? And of course, this is a difficult problem, but our time has been really put on, especially as a NASA postdoc, I did this really then, that how do we create this experimental system and eliminate all the question marks that come from addressing these difficult questions. So the challenge is then how do we reconcile molecular systems biology at the rock record? So again, this sort of goes back to the geology biology integration problem, but you will see how we also sort of separate them as two different records in order to finally address this challenge. So I developed this sort of framework after tackling ancient translation proteins and E. coli system, doing sort of Lansky style experimental evolution experiments, that this is not an easy task, right? So we have to be very careful with what sort of enzymes we pick if we want it to tell us something about Earth history. Of course, if you're interested in this enzymes evolution, there's a vast sea of problems and proteins to pick. But when it comes to connecting the enzyme function to planetary scale behavior, I think that there needs to be certain checkpoints that one needs to visit even before putting in years of time to resurrect and study these things in depth. So I think this is where I am right now. It may change in the future, or get more complicated, but I think there are three main criteria that the system needs to first address before the researchers put in the time to reconstructing the ancient biogeochemistry in the lab. One is that ideally you would want a geologic history to connect eventually to connect the behavior of the enzymes that you're going to study. So this is why a carbon record is great for this, because it's one of the oldest records of life. There's of course the sulfur, there's the nitrogen. These are the first ones that come to my mind. And there are many, if there's a presence of the sort of fossil geochemical record that one can relate to protein function, then you hit the jackpot. That's why I think Rubisco is an excellent system for these studies, because it is thought to be such an important dominator of the carbon isotope output in general. And the next thing is the phenotype, right? So we ideally want to study these things in the lab. We want to go from in silico analyses to laboratory work. Is there some sort of phenotype that we will be able to generate that is characterizable and geologically relevant? What are we actually measuring here, and how far can we take this in our studies, and what are we trying to tie this to? So even though the geologic history and phenotype may seem overlapping, and they are, I think you may also be realizing the nuance, the difference between these two when it comes to studying experimental work. And finally, we want an appropriate modern host organism to study, right? So we don't want to engineer a spinach if we are interested in deep time, right? So we want to be able to engineer organisms that we think existed. That's why, for example, cyano is great for this, that we have some sort of geologic history. There is a characterizable, geologically characterizable, even distinct phenotype that can be found in the rock record. And there are a lot of engineering systems that we can study using modern cyanobacteria today. This is why also I think the sort of tools that come from modern biology today, it's just been remarkable. I think that I don't think biology ever been this fascinating in the sense of the things we can do. It's sort of begging for us to go and use them to answer these questions again about early life evolution. So we are starting with molecules. So how do we so let's just take a few steps back. How do we create these sort of ancient DNA sequences? We start by picking our protein that ideally checks these sort of checkpoints that I just presented to you, part of our framework, if we are interested in the ancient biogeochemistry. And then we start by protein sequence alignment, and this can be a step where you quit and switch to something else if your alignment is not well. So the quality of the alignment in general impacts the quality of the tree that you're generating. So a lot of gaps can be a problem. Lack of representation of sequences from diverse taxa is a problem, and you don't want to bias your sequence alignment to a certain branch, because then the ancestor will reflect that branch that is highly represented. So one has to be quite careful in this sort of seemingly easy step of collecting DNA sequences and aligning them. And we move on to phylogeny. This is the standard sort of phylogenetic step at this point. There's nothing too different than any other phylogenetic work, except that we generate species tree and as well as the protein tree and the DNA tree, and to compare all the topologies to make sure that we are consistently getting the same results. And then we reconstruct the ancestral node by synthesizing this ancient DNA. At that point, it's either cloning this gene inside a plasmid and expressing that in the lab, or what we've been doing is mostly sort of engineering these ancient DNA sequences inside the microbial genome, and then see how does the entire microbe respond to this foreign component. And the sort of one additional step we introduced to this is then to connect the behavior of the organism to biogeochemistry to allow, or at least overlap our results or compare and contrast rather to the biogeochemical data set that is on hand. So why do we go through all this trouble? I will just take a very quick step back, and then we will zoom right back in. But so you're looking at this, it's not even up to date. There's a lot more, but we now confidently know that there are thousands of planets outside of our solar system that are promising, and promising in the sense that they may have same Earth size, or the temperature may be similar. They may have a moon system, not quite that what we have, right? Our system seems to be quite binary compared to what we see, but there are a lot of planets and moons out there that are pretty promising. So astronomers are every day finding even more exciting planetary data sets, And this is why I think this is coming to us, to biologists. It's going to be getting difficultly hard to ignore, I think, for biologists to tackle these problems. But also, when it comes to understanding the habitability of a planet, it's not just about the size and the age or the temperature of the surface temperature, or even other components that are impacted by geology. But there are other sort of, in a way, determined factors that are interesting for biologists. And one of them is relating itself to elements and the entire metalome, perhaps, that could potentially be present on this planetary system that we're looking at if there's some sort of life that is similar to ours. So this inevitably allows us to ask, okay, then what really determined the sort of the metalome that we see on our own planet? Was it because there was a lot of environmental abundance on our planet itself across history, or was it the evolutionary forces or the functional properties of the cells? And of course, yes, metabolisms, and you know what I'm going to say, enzymes, that determines the selection of these metals. And this is, of course, a pretty old problem for Earth system science. It's a fundamental question for this field. That we care about this. We know that the elemental abundances that we have on our own planet impact not only the presence of life or the diversity, but also distribution of it across the planet itself. So here you're looking at the areas of high chlorophyll that are very biologically productive because of an abundance of different nutrients in these certain areas. So the question is then, how did these dependencies evolve? So we asked this question to explore the natural selection of the chemical elements during the coevolution of life and the environment, to specifically address what drives the natural selection of the chemical elements in biology. That is the functional availability, or is just an historical accident. Of course, there were multiple different metabolisms that we could focus on. We focused on the nitrogen records, in particular nitrogenases and nitrogen fixation. So why did we do this? And I just want to give a quick review of the metal abundance and biology, or what we've seen on our planet so far. This is a sort of the figure that allows us to sort of take a quick view of the change of the Earth's surface redox evolution in a way, or the change of oxygen amount throughout the history of life. It's sort of one of the most cited, I believe, papers in astrobiology as well, by Tim Lyons. And so as you see, we don't have any, and we know this, we don't have oxygen about the first 2 billion years of life on our planet. And then we have this sort of whiffs of oxygen that we think is present early on. And then the great oxidation events taking place, and stabilization of the atmosphere, followed by a secondary oxygenation. So the question is then, how did the metals change across this time? And I've been playing with this yesterday, so I hope the animations work. But we know that these elements that were present weren't necessarily in the same amount, and their amount also changed over time. So one of this, as you can see, is the molybdenum that originally isn't in high amount, but then the amount increases. And as we come closer to the present, their concentration also changes. So what I'm trying to show with this sort of animation is that the elements themselves aren't necessarily represented in the same amount across time. And geologists are very familiar with this. Of course, geobiologists are very familiar with this information. But yet, at the same time, when we try to understand how the proteins are binding to certain metals today, I think in terms of biochemistry and biochemical exploration, we don't really put this in the context that these elements alone, we don't even know why they are selected at first place by these enzymes that are very essential. So the questions then are, which metals did early life use, and is the choice of metals driven by the metal availability? So we rooted our, I'm sorry, check the time. Okay, we rooted our problem to nitrogen fixation, at least for this particular study. So I'm going to spend the next few slides sort of showing you our attempt and how we reconstructed the ancestral nitrogenase and characterized it. And this work was led by a wonderful postdoc in my lab, Dr. Amanda Garcia. So we started with exploring the nitrogenase evolution as just a standalone protein system. And our goal was to explore the origin of nitrogenase and then also engineer, not only reconstruct the ancestral forms of nitrogenase enzyme, but also develop an experimental system to engineer this gene inside the modern microbe. And I'm not sure if he's online, but I really am grateful to Dennis Dean and Valerie Kesh, who opened their lab to us for us to go and visit and learn how to tackle with the azotobacter system to engineer it at the genomic level. So nitrogenase also is an interesting problem. The origin of the enzyme is not quite known. It's a sort of a broad, I represent this as dash lines, question mark there. It's not well constrained, maybe relatively compared to other systems. But that didn't stop us because we just really want to understand if we give it an attempt to reconstruct the ancestor, what does the ancestor do and what sort of metal dependence does the ancestral version have? So this is a very, of course, important enzyme. I think requires an introduction to many, but in this audience perhaps, but just to, as a refresher, we're looking at sort of life's way of making the atmospheric nitrogen biologically useful for itself. And it comes down to this enzyme that is metal, it's a metal enzyme, but also it's quite interesting because it's a pretty sort of metal enzyme dream that it prefers different, there are different versions of this enzyme that prefers different metals from molybdenum to iron, and these are all modern homologs. So the obvious question, of course, is that how did the nitrogenase metal dependence evolve, and whether what would be observed if we were to reconstruct the ancestral variants of this enzyme. So there were some studies that we built on that characterize the modern homologs and try to understand the representation of different metals and looking at amino acid sequences as well. So we had a lot of prior published data and knowledge that we could use to interpret our own findings. So the question was then again, if the metal availability of molybdenum, for example, that's the more abundant form of nitrogenases preferring molybdenums, knowing that the amount of molybdenum is low in ancient oceans early on, but yet then there's an increase of molybdenum, was this also reflected by the enzyme itself, looking back? So it was quite a simple question, actually. And you may think that, of course, if the molybdenum amount wasn't high early on, then enzyme will not prefer molybdenum. But then when you look at current biota, you find that no enzyme itself is preferring molybdenum. And by sort of the sort of dogma, in a way, I think we have in our mind, dictates us that, you know, we have to connect these things to environments, right? So, of course, there's more of molybdenum today because molybdenum is higher. You may think. So we started from there. I want to understand whether this molybdenum scarcity early on is reflected on the enzyme behavior as well. And moved on to reconstructing the ancestor using phylogeny to understand the metal dependence. And we did a lot of modeling here with the nitrogenase structures and characterized the metal binding sites and active site structure and sequence. Again, this was all built on the prior nitrogenase work that dictated some of the amino acids as being essential for molybdenum binding or metal binding, or the active binding pocket site being important. So we sort of compared our own analyses with the prior work in that sense. So we built our tree using concatenated nitrogenase protein sequences. As I believe Douglas mentioned also, there are multiple genes. This is a sort of rather big enzyme in the sense that it has multiple domains. And we created a phylogeny tree collecting as many sequences as we possibly can using today's nitrogenase sequences. And it was nice because we could see some nice bifurcation, that the anaerobic lineage and the aerobic lineage is bifurcated nicely. And then we have more diversity on the sort of upper part of the tree here. So we zoomed in because that's sort of what we see an interesting metal dependence, that we have the diverse organisms that have vanadium or iron. And as we sort of travel back further from ancestral AIDS ANC to ANC and eventually here ANC, we wanted to basically focus on these particular nodes and then reconstruct them and understand their metal binding properties. by using the sort of inferred sequences overall. And this was the first sort of step that we did. This is a published work, so I'm not going to drill down too much also for the sake of time, but we knew that there are certain amino acids such as cysteine and histidine that are important for the sort of the anchoring the metal itself and the cofactor itself. So this is all what we do. So in general, we look for the presence and the conservance of certain amino acids and domains in these ancestral forms, especially in the beginning when our experimental setup may not be ready to characterize them right away. And we are a young lab, so most of the time, yes, the experimental lab is not ready. So we try to understand as much possible by looking at the sequences. And what we found quickly is that the ancestors in general retained the conserved active site residues, and some of them are more specific for a certain metal binding property. All of these sequences and the trees are available in this address, and the slides are posted and this talk is recorded. So feel free to download them, use them. And if you don't have any access for whatever reason, please email me. We want the community to use these ancestors. So especially I've been alerted that if someone is interested in using ancestral nitrogenase sequences, we are more than happy to provide these things to you so you can study them as well. And we moved on, and I will move rather quickly now. I'm worried about time. Let me check I think, okay. All right, so we focused on analyzing the pocket volume that was previously by Sean McLean and Victoria Orphan was used to classify the metal dependence of these enzymes. So this is a lot of structural prediction and calculation of the metal binding pockets. And at this point, we couldn't really assign the metal binding pocket as a sort of a proxy for the metal specificity. Unlike the previous published work, actually, we think that the pocket site may not be a good indicator of the metal dependence. So it was quite ambiguous, at least for our analysis. Then we drilled down a little further and looked at the sequence to understand the active site sequence composition and see whether there will be any correlation with the metal usage. So, and we know in today's this biochemical work, and there will be, I think, a talk tomorrow, or maybe 2 talks about this tomorrow as well, that the active site residues can discriminate the iron, vanadium from molybdenum. So we compared a lot of comparison here with a lot of, we compared thousands of sequences to understand whether what does the oldest ancestor resemble. And what we found is that the oldest ancestor resembled molybdenum more than the iron, vanadium, which suggested that the oldest ones actually were preferring molybdenum, at least by characterizing the sequence features. So we then took this just a little step further and tried to understand what then allows the metals to be present on this enzyme. So of course, it's not just the enzyme, there's all these assembly proteins that help the enzyme to put the metal site on it in the simplest way I can put, really. So we know that this sort of nitrogenase cofactor biosynthesis requires additional enzymes. So then we tracked the evolutionary history of the cofactor biosynthesis enzymes and try to understand whether they're present across the tree or whether there's a time where we lose them. And what we found is when we compare this, particularly the NIF-E and NIF-N genes that help the assembly, is that while the vanadium, or if we go back to the molybdenum branch, these assembly proteins are present, evolutionarily we seem to lose them, especially when we come to the taxa that is, for maybe some people, is not even a nitrogenase, it's more like a nitrogenase-like taxa here, that we don't see any assembly protein as we go deeper in the branch, in the tree. With that, we concluded that the ancestral organism may also lack this assembly proteins. So this also makes an interesting, opens an interesting problem that the oldest ancestor uses molybdenum, but does not have the complete metal assembly pathway. So of course, then what does this mean in terms of the presence of the metals, and how did this ancient organism use its own assembly to help the nitrogenase or something like nitrogenase to carry out nitrogen fixation. So in summary, the sequence features, even though they represent molybdenum-dependent ancestry, the oldest ancestors lacked a full cofactor biosynthesis pathway. So we are going further on this, that we think that maybe there may be an alternative or a simplified pathway for ancestral molybdenum incorporation. I will be very interested if you know, looking into the more broader metabolisms. Of course, this is something that also may be interesting to some people in this audience, but we're interested in also looking at the uncharacterized nitrogenases and organisms that host these enzyme variants and see what sort of pathway they might have. And what, as a first step, we've developed this experimental system to bring back these ancient nitrogenases, to study them using Azotobacter model system. This again went back to our framework and checking all our factors of what we need to have in order to reconstruct this deep time in the lab. And we published this protocol so that you all can use it, but we are able to, definitely we were able to reconstruct a sort of a younger ancestor in the lab, but lately we got a Precambrian age ancestor as well, and the organisms are, you know, it works. So we're able to actually reconstruct this ancient version inside the modern organism, and we're doing quite a bit exciting work using this hybrid organism now. But I'll just also mention that we've reconstructed the entire synthesis pathway and looking at their ancestry and see how the sort of variation across different trees that we create impact our analysis. And this will be available in a couple of weeks. I will post a preprint on this, that we think that the ancient nitrogenases or their predecessors going further backwards may not even be a nitrogenase enzyme. So, so with that, I'll just want to visit this sort of main-ish finding so far, that the ancient nitrogenases likely prefer molybdenum. So what does this mean? I think this was, again, quite interesting because especially when we think about the Precambrian, I think that there is really value in thinking that the protein level innovations and the sort of molecular innovations shaped the biogeochemical cycle, as opposed to maybe organismal level innovations that we see that dominate the Phanerozoic. So I think really there's real value in digging through the molecular innovations when it comes to understanding the Precambrian life all the way into the past. And definitely enzymes are into this, in a key part in this. But one thing that I want to sort of leave the audience with as a final note is that even though our attempts are really focusing on integrating the molecular and geological record in many ways that I also tried to present here, there may be some value in decoupling and these two records first. And by that, I mean really treating them as their own independent records before sort of forcing ourselves to integrate them, and before interpreting everything we see, just making these quick assumptions about what they may tell us about the past, that they exist in the same volume, but they're independent and in many ways, and they maybe the trajectories can be decoupled in some key points. And we shouldn't judge the biology by the geologic box and resist this urge of connecting these reservoirs before truly understanding them as their own data sets. So, and with that, I will wrap. Of course, we are moving forward to understanding and connecting these things at the planetary scale, as I mentioned in the beginning, and we want to understand whether, you know, what if we are confronted with some elemental data or any molecular data that gives a sort of a promising information about, okay, the presence of a potential nitrogen cycle on another planet, what will take us to conclude that this may be biological or not. And to explore this question further, we just got rewarded a NASA center. We named it MUSE. It's the metal utilization and selection across eons. So I have the honor to lead this for the next 5 years or so. We will be exploring the natural selection of chemical elements in many different ways. We are starting with nitrogen, but we're not going to stop there. And there's going to be a lot of exciting projects here. We are hiring, so I just wanted to mention that as well. And I will wrap right here. Thank you so much for having me. This is quite an honor. I really wish that we could, hope that we could also meet in person, because I can see definitely very nice overlaps between our work between MUSE and ENIGMA. So thank you so much.
Thank you, Betul. That was a wonderful talk. I'm sure there are lots of questions. I know I have at least one, but we're slightly over time and we need to go to the next speaker. But there is a discussion at 2:30 to 3, and I would be very happy, or we would be very happy if you stay and take some questions then. Right. Our